Okay, I'd like to thank the organizers for inviting me and letting me see how wonderful Sydney is. So I am going to focus this talk on the transcriptional regulation of cardiomyocyte maturation. So we've already heard many times how myocyte maturation is a key issue now in regenerative biology. So if we want to use iPS cells to make disease models or for replacement therapy or even for fibroblastic cardiomyocyte reprogramming, we're faced with this problem that the cells that we make usually have a phenotype like this, a very immature fetal-like phenotype, whereas we would like to make mature cardiomyocytes. And you could refer to this as a bottleneck that's inhibiting our ability to pursue regenerative cardiac biology. So first, let's just go into a little detail about the differences between immature and mature myocytes. If you compare fetal to adult myocytes, they have different transcriptional programs, particularly in sarcomere isoform expression, obvious differences in size. In mice, it's about a 30-fold difference in size between a neonatal and an adult myocyte. Nucleation is in rodents, fetal myocytes are mononucleated compared to binucleated adult myocytes. Fetal myocytes grow by mitosis as opposed to hypertrophic growth of adult myocytes. The fetal myocytes have a glycolytic metabolism compared to oxidative phosphorylation in adult myocytes. And we like to look at T-tubules because these are a marker of definitive mature myocytes. So we're interested in how all of these different aspects of myocytes are regulated. And if we group them into 4 different categories of phenotypes, how are they coordinately regulated? Is there a central transcriptional program that's controlling this process? So anytime you want to study how some gene Y regulates a process, we typically use gain of function and loss of function approaches, often with different mouse strains that have either transgenic expression or knockout of the gene. But we all know that this is very time-consuming. You need to get a lot of mice and breed a lot of mice. Some phenotypes are not accessible due to lethality. They're not amenable to genetic screening. And I think that this is really one key limitation that often when you manipulate a gene in an organ, you'll get organ-level phenotypes that really confound your analysis of the cell autonomous function of the gene. So that's just shown in more detail here. If we manipulate a gene across an organ, you get the direct effects, which cause the cell autonomous phenotypes, which then cause a feedback loop, so that you get, by the time you make your measurements of downstream phenotype, it's quite distal from the actual manipulation. So we like to pursue a strategy in which we purposely make a genetic mosaic in which only a small percentage of the cardiomyocytes in the organ are modified. We use AAV to create these genetic mosaics because this is a non-pathogenic virus that can efficiently transduce cardiomyocytes. So here, for the gain of function, it's easy. You put the gene in the AAV and you give it at a low dose, you can get a genetic mosaic. For the loss of function, we use a combination of AAV and CRISPR-Cas9 to create somatic mutants. So here, just to I'm sure everyone's familiar with Cas9. It's a nuclease that's directed to its target by a guide RNA, causes a double-strand break that can lead to an indel. And two-thirds of the time, that'll cause a frameshift mutation. So there was a nice mouse model described in which Cas9 is activated by Cre in the Rosa locus. We deliver AAV that expresses Cre specifically in myocytes to activate the Cas9, which also has a GFP label. The AAV also expresses a guide RNA, which then directs the somatic inactivation of a target gene. So we purposely give these viruses at a low dose so that you only get direct inactivation or overexpression of the, in a low percentage of cells, so that we can focus on the single cell phenotype and avoid the confounding effects of organ dysfunction. So as an example of this, I'll show you how we studied T-tubule maturation. So T-tubules are these invaginations of the plasma membrane in cardiomyocytes that you see at about P14 in mice. So these invaginations are important to help to synchronize the excitation-contraction coupling across myocytes because they're so large. And again, this is a hallmark of a mature myocyte. So there's actually very little known about how the cell makes T-tubules. One of the key players was thought to be this gene junctophyllin-2, JPH2, and that was largely based on some work in which a transgenic mouse expressed a shRNA that knocked down junctophyllin-2. And this caused loss of T-tubules, so it was inferred from this experiment that junctophyllin-2 is essential to make T-tubules. I think that's a reasonable inference. But there was, again, a confounding effect in that these mice had heart failure, very low ejection fraction. And it's known that that itself will cause disassembly of T-tubules. So we took this as a proof of concept study where we could use a somatic inactivation of junctophyllin-2. It's stuck. Okay, so here we fact sorted the GFP positive cells, and then we performed a Western blot for junctophyllin-2, and you can see that we had very efficient knockout of the gene in the GFP positive cells. Now again, our goal here is to do a genetic mosaic, so we did a dose response of different doses of AAV to see, to transduce different percentages of cardiomyocytes. At a low transduction efficiency, we didn't affect heart function, but at a high transduction efficiency, we had very bad heart function. We then did in situ imaging. So here we just take the heart right out of the mouse and put it on the confocal stage and take optical sections. So here at the high dose, you can see that this GFP positive cell has a very poor T-tubule morphology, consistent with the idea that junctophyllin is important for maturation of the T-tubules. But if you look at the adjacent cell that's GFP negative, it also has bad T-tubule morphology. Now, if you look at this low dose, this GFP positive cell to the naked eye looks like it has normal T-tubule morphology. So we quantified this effect, and again, at the high dose, there was a strong effect on T-tubule morphology, but you'll notice that even the GFP negative cells had highly abnormal T-tubules. This points out the cell non-autonomous effect of organ dysfunction causing this T-tubule defect, and in that setting, there is an additional effect of the junctophyllin knockout. But in the low dose, you can see that there was a tiny effect on T-tubule morphology with junctophyllin knockout, but that was not visible to the naked eye. So to summarize this, I'm basically showing you this to explain why we take this mosaic approach, that you need to do a genetic mosaic in order to study the function of genes in maturation, because when you manipulate them across an organ, you will automatically induce maturation defects that may have nothing to do with maturation. So just about junctophyllin, we conclude that it has a minor role in the formation of T-tubules. Its depletion to a high percentage causes heart failure, which then precipitates additional destabilization of the T-tubules. And just as a more technical point, we think that this genetic mosaic approach is really essential to study genes and their role in maturation. And without it, it's very difficult to circumvent the confounding effects of heart failure. And we think that this Cas9 AAV platform is a really nice way to inactivate many genes in a mosaic pattern. So we use this strategy to study the transcription factors that regulate cardiomyocyte maturation. Here we picked a panel of some of the best-known cardiac transcription factors, and we individually targeted them for somatic mosaic knockout. So these 8 factors as individual knockouts had no effect on T-tubule morphology. But this gene, SRF, when you knock it out, it causes a severe disruption of T-tubule morphology. So we looked into this in more detail by getting the floxed mouse, and so now we're making the mosaics by giving a low dose of Cre. And this is what one of these SRF knockout cells looks like. It's very long and skinny and has a small area, and it has essentially extremely disrupted T-tubule morphology. We also did this experiment by giving the virus in adult myocytes. And what I hope you can see is that when you give this knockout SRF in adult myocytes in a mosaic pattern, there's almost no effect on the size or shape of the cell or on its T-tubules. So again, we looked at size as well. In the neonate, there's a strong effect on size, whereas in the adult myocytes, there's a very small effect on size. So from this, we concluded that SRF is a stage-specific regulator of cardiomyocyte maturation. So we wanted to look more into the transcriptional role of SRF in the different stages. So we did RNA sequencing in the neonatal knockout versus the adult knockout. What I'm showing you here is that when you knock the gene out in neonatal cardiomyocytes, you have a large effect on many genes, almost 1,000 genes. Whereas in the adult knockout, there's really a very small population of genes that are affected. And if you try to do a correlation of the full change of the genes in the neonatal knockout versus the adult, there's very little correlation. So this again points to a stage-specific role of SRF in regulating these genes. So we looked at SRF has been studied a lot. Some of its best-known targets are the sarcomere gene isoforms. So here's a list of sarcomere genes in the neonatal and the adult knockout. And what you can see is that many of the sarcomere genes are aberrantly expressed. In particular, in the neonatal knockout, you get a downregulation of mature sarcomere isoforms and an upregulation of the mature isoforms, of the immature isoforms. On the other hand, if you look at the morphology of the sarcomeres in the neonatal knockout, you can see these minor defects in sarcomere structure, but certainly not what was described previously in the organ-wide knockout. And then in the adult knockout, the sarcomeres are essentially normal. So we did chip sequencing with a biotinylated allele of SRF to look at the differences in chromatin occupancy that might account for this stage-specific gene regulation. And once again, it's not. So once again, we found that in the neonatal mouse, SRF occupies many more sites than it does in the adult mouse. So the majority of sites are lost in the adult mouse. And you can see that more graphically in this tag heat map display. So we think that the stage-specific role of SRF in regulating sarcomere gene expression has to do with its changes in chromatin occupancy. If you look at the Go terms of the differentially expressed genes, you can also see that many of the top Go terms were related to mitochondria or metabolism. And some of the chip sequencing regions show that mitochondria-related genes are direct targets of SRF. So from this, we concluded that SRF actually has a direct role in regulating mitochondrial biogenesis, which is a function of SRF that was not noted previously. So just to summarize some more about this part of the talk, we showed that SRF, I didn't show you the GATA part, but SRF regulates cardiomyocyte maturation. This is in multiple aspects of maturation, including the size of myocytes, sarcomere isoform expression, metabolism, and T-tubulation. SRF regulation of these features is specific to maturing cardiomyocytes, and that the stage-specific SRF chromatin occupancy accounts for this stage-specific regulation. So we just returning to this picture about cardiomyocyte maturation. So now we have SRF as a key regulatory factor, but we're interested in understanding the relationship of these different aspects of maturation. So for example, are they linearly associated with each other in a pathway like this? Or maybe they're in a bifurcating pathway like this. So we'd like to understand which of these, if there's a hierarchy of the regulation of maturation. So again, we use this somatic mutagenesis system to systematically mutate genes that are related to each of these phenotypes. And I'll just skip the raw data and show you the conclusions in this table. So if you knock out sarcomeres by knocking out MYH6, you can see you get very small cells that have a high length-to-width ratio and disrupted T-tubules. If you knock out T-tubules directly by knocking out RYR2, you get very small cell area and, of course, disrupted T-tubules, but the sarcomeres and the mitochondria are normal. If you disrupt mitochondria using mitofusin 1-2 knockouts, or we've also done TFAM knockouts, or dominant negative DRP1 expression, surprisingly, you don't affect any of these aspects of cardiomyocyte morphology, which is sort of opposite to the conclusion that Enzo presented previously.
William, we've got about probably 1 more minute.
Okay. So this is the summary of the hierarchy that we've established. So SRF, we think, primarily regulates sarcomere isoform expression, which then affects T-tubules and maturational growth. And on the other hand, SRF also regulates mitochondrial metabolism, but these are not, it's not linked to these other phenotypes. Okay, in this last minute, I'll just tell you very briefly that what we're really excited about is that you can use this Cas-AAV system to do forward in vivo genetic screens. So we've mutated 2,500 transcriptional regulators and then screen the single cell phenotypes using this developmental reporter, MYH7 YFP. So we developed this library of AAV. We delivered it to mice and then essentially just did fact sorting to measure YFP expression. And in the YFP positive cells, we sequenced the guide RNAs to find the guide RNAs that are enriched in the YFP positive cells. So this just shows you that the positive and negative controls worked. But here's the result of the screen. So here we have all the genes ranked by their p-values, and these are the ones that have significant p-values. So we validated the top 10 of these hits. THRA is the thyroid hormone receptor, which is well known to regulate cardiomyocyte maturation, but here's a lot of novel genes that have not been previously implicated in regulating myocyte maturation. So I'll just summarize this and say that we're excited about how this AAV mutagenesis platform can be used to do forward genetics. So here we've done a forward screen for cardiomyocyte maturation, but we envision that this screen could be used to look at hypertrophy, proliferation, and survival. Thank you very much.
Looks like we're going to have some competition for questions here. Off you go, Enzo.
Hi, Bill. Great talk. Just to clarify with your comment about reconciling our data and your data, we've shown that metabolism is a key regulator of cardiomyocyte proliferation, but we also showed in our paper that altering the metabolism of the myocyte did not affect the structural or electrophysiological maturation of the myocyte. So I think our data might actually be consistent with your findings in that regard. And I'm wondering whether you've looked at cell cycle regulation in your maturation experiments there.
Yeah, so we have looked a bit at proliferation in the mitochondrial knockouts, for example, in the TFAM knockouts. We didn't really see much increased proliferation in those cells, but I think I think it's totally reasonable that we get different results in that we're looking at what's required for maturation, morphological maturation in vivo, whereas you're looking at what's sufficient to drive maturation in an in vitro system. So it's a different question.
Terrific. I think just in terms of the interest of time, we're running a little over. We might move on to the next speaker, but a big round of applause. Thanks very much, Bill.